Today I'm going to walk you through the protein production Jackpot lab from Kansas corn. Hi, my name is Bill, and I've been teaching for 30 years. I currently teach at Caldwell High School, and we're going to go through a protein production Jackpot lab. I'm going to show you some of the ins and outs and keys to success for getting good results for the lab. Okay, to start with this lab, something we're going to look at the objectives first of all, and that's we're going to try to extract the protein, a specific protein, the PGlow protein, in fact, from an organism. Okay, we're also going to learn to use column chromatography, and then we're also going to extrapolate how extracting a desired protein can be beneficial to humans. Okay, so to start with, we've got a PGlow plate that's already been set up. So the PGlow lab activity is a lab activity where we introduce a gene into a bacteria, and to determine if the gene's present, it produces a fluorescent green glow. And I'm not sure if you guys can see this, but these big white colonies here are glowing green. The smaller colonies, you can see a bunch of little white colonies in there, they're not glowing green. And that indicates that the green colonies have the gene in them, and the white colonies do not have the gene in them, okay? The gene product, at least as far as that goes. So what we're going to try to do, so we could transfer a gene into a bacteria, all right? We've successfully done that with PGlow. What we want to do now is take that protein out. And I mean, if you could produce a protein that can be beneficial to others and humans, we got to be able to get the protein out. And this is how we're going to try to get that protein out. Following this procedure from this protein production Jackpot lab. So again, materials you're going to get with all of this, the main, I guess, instrument you might need would be just a microcentrifuge. You can use a regular centrifuge with just microtubes in it, okay? That still works. An incubator would be great, but you can incubate at room temperature. And then the other aspect is having some kind of black light, and in the kit we provide this black light, actually one for each group, so they can determine if they've had successful results, okay? So we're going to go through the quick setup on this, and this is all based on the assumption that you're starting with a plate that already has transformed bacteria, okay? So the PGlow is on this. I'm going to pull some of the bacteria that are glowing. I want to get some of those bacteria off, and then I want to get some of the white bacteria that have not been transformed. And we're going to use those as a kind of a control. I need to try to get the bacteria on this loop. Now, I'm not talking about digging into the whole agar. I just need to get some surface bacteria. Before I get that, though, I've already transferred 2 milliliters of a nutrient broth into these 2 test tubes. One test tube's labeled plus, and the other test tube's labeled minus, okay? So this is going to be the positive one, and this is going to be the negative one. The positive ones are going to be the green ones. So for me to get the green, and again, you can actually see that it's the large colonies that are going to be green. So I'm not going to necessarily have the light on it the whole time. Your students might need to have somebody shine the light on it, because I want to get a green colony on this so that I could put it into this container, okay? So I'm going to very gently get just some green colonies, and I don't need to get a lot of it. And I'm just like to get a little extra just in case. Put the lid back on that as soon as possible, because that is a culture, a bacterial culture. I'm going to put the loop in here, and I don't want to touch the sides. I wanted to get it into the liquid, and then I'm just going to spin this loop around, trying to get all that bacteria off that. So you do essentially the spin. And again, the assumption is that that bacteria will eventually get off that little tiny loop, okay? Now the other aspect is this is bacteria on it, so you do not want to put this just down on the table because it's got active bacteria in it. So you've got a trash container or some kind of container where you're going to put that to where people won't be exposed to it. So that's the positive. I've got the PGlow positive bacteria in that. The negative one, I'm going to try to get some of the white bacteria. And then once I've got that in there, we'll let these tubes incubate, okay? So to start with, I have to get another different, completely clean loop, sterile. And this time I'm just trying to get the white colonies. And again, without getting any of the big colonies. And again, the idea would be a small amount. That they say in the instructions, smaller is better in this case, because if you get too much, you may not get to the actual protein if it's present. Now this one's a negative, so hopefully we're just getting negative bacteria. Again, same thing. You want to put that loop in a container that will essentially keep it from other students getting exposed to it. So I don't need this plate anymore. I'll put this out of the way. So according to the instructions, after I get these two set up, right now the only other thing I'm going to do, and what I can do, by the way, to verify that I actually got something in there, I don't know if you could see it, but there is a little glow in there. And this is the positive, and this is the negative. And I don't. It's a faint glow because I've only got a small amount of bacteria in there. So what we're going to do is we're going to put this in a broth culture, and we're going to let the bacteria grow up. Okay, and after it grows for a day in an incubator, we'll have a lot more bacteria in there, and then we might be able to see some more of that glow. Okay, so according to the instructions we're going to be using, we're going to incubate this at these two tubes in an incubator at 37 degrees overnight. The other option, if you don't have an incubator, is to leave these out at room temperature, and at room temperature for 2 days. And that'll be enough time for it to actually get enough bacterial growth where you can get some of those proteins produced. Okay? So that's the incubation time. Now, for what we're doing today, I've actually done that already. Okay? I've actually incubated another set of the exact same bacteria from the exact same plate. And I've got those tubes right here. So in this case, what we're going to do is very simple. After those have been incubated, we're going to take 2 mLs of this incubated bacterial solution and put them in 2 small microtubes that I've labeled plus and minus. Okay. So to do that, we're going to be using these disposable barrel pipettes. And these barrel pipettes have different marks on them. It just so happens that, I'll do the negative first. It just so happens on this pipette, this mark right here indicates 1 milliliter. That would be 0.75 milliliters. That would be half a milliliter, 0.5. This would be 0.25 milliliters, etc. So this is one way we're going to use it. We don't have to get a special micropipette out. So I'm going to take the broth culture that has been incubated. I'm going to get 2 mL. So to get 2 mLs out of this, I'm going to put 1 mL and do that twice. Once I have that done, that one's finished. As a general practice, this is in bacterial culture too, so again, this would also want to be put in a waste container, not accessible to others. And again, the last one I'm going to do here is the positive. Do the same thing. I'm going to transfer 2 mLs of that into this microtube, and then we're going to try to spin this tube down to see if we can actually get the bacterial pellet there. Okay, so I'll take okay. So again, once those are done, the next step in the procedure is going to be spinning these down. The goal of spinning this down is to get all of the solid bacterial material to the bottom to where we can actually just use that, not any of the liquid that's left in there. So this will run for 5 minutes. And at the end of the 5 minutes, what we're going to do is take those tubes and use just the pellet that's in the bottom of the tube. Okay, so we've spun this down for 5 minutes. Okay, the tubes that have 2 mLs of the broth bacterial culture solution. I'm going to pull these two out real quick, and we're going to take a look at them. This is a tube that has negative, is the negative that did not have, supposedly did not have any of the glow material in it. And if you can tell, there's no glow in that. There's a little tiny pellet at the bottom of that tube. And if you look at this tube, you see a little bacterial pellet with the glow, okay? And that's our target. That's what we want, okay? So, according to the procedures, okay, what we're going to do very simply is the supernatant is the liquid that's in this, okay? The pellet is the solid bacterial sludge at the bottom, okay? Solid material, okay? We're going to pour out the supernatant, okay, and leave just the pellet in there, because that pellet has the protein that we're going to try to target, all right? So, that being the case, we're going to now, since there's no glow bacteria in this one, the negative one, we're not really going to process this. We're simply going to work with the positive one that has shown that it has a glowing bacteria in it, okay? So I'm going to pour this off very gently. Now again, if you pour it off too fast, you might pour the pellet out. That's not a good idea. Generally speaking, what I try to do is gently pour it slowly down, trying not to dislodge the pellet. And there might be a little liquid left in there. Not too bad of a deal. How do I know if I've just got the pellet in there? That's mostly just pellet with a small amount of liquid in there, okay? Not a bad deal, that liquid in there, okay? So, now according to the procedure, what I'm going to do is I'm going to add 250 microliters of TE buffer, okay? 250 microliters. We don't have a micropipette. We've got our trusted disposable pipettes, and these have a mark on them for 250 microliters. Remember, this was 1 ml, 0.75, 0.5, 0.25 mLs is 250 microliters. So that's the mark I'm going to fill this up to, and then I'm going to put that into resuspend this liquid, okay? So that obviously isn't a perfect mark there. So if you get close to it, you'll be fine. I put the 250 microliters in there, and one thing you don't want to do is drop your cap. Now, again, according to the procedure, you can rinse this one out. So when you distribute your equipment to your lab stations, they don't need 15 of these. They can actually get by with 4 of these for each lab station. And this last one we can just keep rinsing and reusing, okay? So I'll keep that out here because it's used with the same tube each time, all right? So I've got this liquid in there, okay? Now I want to try to make sure that bacterial pellet gets mixed up in this, okay? Before I mix it up completely, I still need to add 1 drop of lysozyme, okay? And lysozyme is an enzyme that breaks down the cell membranes, okay? And the cell walls, actually, of the bacteria. And that cell wall is what's stopping any of the material from coming out. So I'm going to use this enzyme to break this up. Pretty simple. 1 drop is what it's calling for. The distribution in a classroom, you'll probably have to have a station set up to where they come and get this drop, or you might actually come by and give them the drop. This is a small volume of liquid you've got in this container, and it might cause a problem if somebody spills it all, because then you would be missing all of your lysozyme. So you don't need much, but again. So we've got an enzyme in there, we've got a buffer in there, we've capped this, and now I'm just trying to resuspend that pellet, okay? And as I resuspend that pellet, one of the things that the procedure asks you to do is put this in a freezer overnight. When you put this in a freezer overnight, any of the bacterial cells that are in there will also rupture from the freezing. And that would be the actual goal, is to rupture that the cell walls of those bacterial cells. And if you can do that, you're going to actually get a good yield, because that's one of our optimal ideas there, okay? So I actually have set the exact same setup earlier a day ago, and I've got that thawed out right now. This is a positive test tube. that went through the same procedure we just did. The only difference is this has already been sitting in the freezer overnight. So I've got it, I thawed it out, and now we're ready to continue with the procedure, okay? So the concept for this is I thawed out. I want to actually get the pellet, okay, back out. So I'm going to spin this. But again, whenever you're using a microcentrifuge, you got to have equal spacing on this. If I put my tube in here, I can't just spin it. It's not going to be balanced. So I got to put a tube with about the same amount of liquid in it on the opposite side so that I can balance it. Okay. And we'll know if I did a good job on this if it starts making a big noise. Okay. So those volumes should be fairly equal, and you want to put them on the opposite. Another way to know which tube, ideally, as taught by using a centrifuge, if you put the hinge side up, when this spins, the bacterial pellet will be on that side of the hinge, so you'll know where to look for it, okay? And then this will spin for 10 minutes, okay? At the end of 10 minutes, we're going to take the tube out, and then we're going to process it and take it through the column. All right, a couple things you can do. If that's a 10-minute spin you just finished, that 10-minute spin is when you can actually have students working on trying to get the columns ready for the column separation, okay? The procedure that's found in the BioRed kits that you get will show you that process, and we're going to go through that process here in just a second. So I spun down this tube, and this is my positive tube, and I've got a pellet on the hinge side, and that pellet's right there, okay? So now I'm going to get this column ready for us to actually use. Now to do this, according to that, you're going to remove the cap, and this cap is right here, okay? And it's easy to do. And we'll snap this off first. Okay, I'm going to put that over a tube, and now I'm going to pull this out. Okay, now there's some residue in there. That's all right, but we're going to be actually pouring some liquid in there to get this tube ready. Okay, so according to the procedure, after you got that cap, ideally we want all that liquid down at the bottom, but we're going to actually use some material to actually make sure it gets down there. So we're going to use 2 milliliters of equilibrium buffer. The equilibrium buffer is going to allow the ions in this chamber to actually be open for the usage when we're trying to separate this. So 2 mLs is going to be 2 of these pipette necks distances there. And I'm doing that by also putting it on the liquid on the side. So I'm putting it on the side so it gets all of that down in there. And that's 1 ml. And that's 2 mLs. So ideally, you're going to do that to help get some of that material down into the column. Okay, so we've got that, okay. After that drains to the bottom. Now, this might take, if you look at this is liquid here, and we're trying to drain this liquid down to the bottom of this. This will take 5 minutes to do. We are proposing, and we've used this quite recently, a way of making this go a little faster is a little syringe with a number 2 single hold stopper, and this syringe can be put in here. And as you see, if I tighten this up a little, actually what I want to do is probably get some air in the syringe first, and then I will actually put this on there. And now what I'm trying to do here, I'm going to try to see if this will work. I'm going to don't know if slowly push some of this liquid through. So I've got this liquid. I'm essentially putting equilibrium buffer through this. And as I get most of that equilibrium buffer, don't forget I put 2 mLs of that through there, okay? And it's essentially helping pack the column down too. So this column has resin that is specific for specific molecules, okay? And that's really what we're doing. We're getting all that in place in this column so that is ready to go, okay? And on the side of this, just to make sure you know, there is a 1 ml mark that we're going to drain that to, okay? And I am almost to that mark right now. I'm just going to go a little further. There we go. All right, so right now I've essentially put the equilibrium buffer through the column. So this is a stopping point, but guys, we're not going to stop. We're going to move through this. They're actually talking about if this is taking time to do this, we're going to use a syringe to make these procedures go through very quickly, okay? So we're trying to get this pushed through the column in a timely fashion versus waiting for it to just drip on its own. So now what I'm going to do with the tube is I've just spun it for 10 minutes. I've got the pellet on the side. I'm going to transfer 250 microliters of the supernatant into a new tube and label it again, okay? So I don't want that pellet because that supposedly is the debris, okay? So I'm going to put 250 microliters of this into a new tube that I have labeled right here, okay? And the 250 microliters, again, is that same volume. This mark right here is the 250. And we're going to put of this tube, so we're trying to get the liquid. The supernatant is liquid, okay? So now I'm going to put 250 microliters of binding buffer, which this solution here is going to essentially bind to the proteins that are in here, okay? So we're attaching a molecule that's going to help bind to the column chromatography. And this, again, is going to be 250 microliters, okay? Use another one on this one. Ideally, if I had a situation where I was in a group, I could have a little rinse container and rinse these out, this pipette each time. I could use the same pipette for this. So I'm going to get 250 microliters. Which is there. This would be another step according to the procedure with Biorad. You can just put this in the freezer and let it sit for a refrigerator. But we're going to move on through this using our technique, okay? So I've got 3 tubes I've labeled right here, 1, 2, and 3, okay? And I'll put them right there in order. And we're going to start working by moving some of this through the column, okay? And again, ideally what we want to do is we've already prepared the column. We're now ready to put the 250 microliters of this into the column. So now what we're going to do is we're going to take and put 250 this solution here into the column, and then we're going to let it go down the side so that it gets into the matrix. Okay, so to do that, I could use my same tube that I just used. That's been rinsed because I'm using the same material. So I'll take the lid off of this. I'll add the 250 microliters. I'm going to add this down the side of the tube. Now we're going to take and put, again, we could, just to verify, you try to verify as much as you can that we do have some glow in there. So some of the material we put in there is a glow. There is some of that. And that's set to go, okay? Now, next step, we're going to take 250 microliters of wash buffer, which is right here. Yeah, right here. And we're going to put that through the column, all right? Again, that wash buffer is 250 microliters, so we're using the same volume for all these liquids, okay? And again, we're going to see if we can get this to go straight into the matrix, because we want that to wash down through the column, all right? Now, we're going to push some of this through. And at this point in time, this tube that I've got in here is, we'll call this test tube 2. In fact, I'm just going to put it into test tube 2 real quickly. So I've got this into test tube 2. We're going to get the liquid matrix to go through there. So we've got this wash buffer. We're washing the proteins that are stuck to the column. We're washing off any excess. And we don't have to get 250 microliters out of it, but that's pretty good right there. So that's going to be test tube 2. Okay, I'll put this back in the original one. And take this off. So the next step is we're going to add 750 microliters of TE buffer, and we're going to let that go through the column. Okay, so 750 microliters of TE buffer is going to go to this line here. And we're now going to put this on test tube 3, okay? And we're going to let this flow through. So the benefit of the syringe here and the whole situation is, if you had to sit there and let this drip by itself, one drop at a time, this is going to take 2 to 3 class periods. I mean, it's stopping points with freezing it, etc. So if you're using this, we should be able to, and again, what I'm going to do is get a little more air in my syringe. And as I do that, we will get the column. So we're getting liquid down here. And that should be most of the liquid through there, because we put in there, don't forget, 750 microliters. Okay, and this is test tube 3. Okay, so we got test tube 1, test tube 2, and test tube 3. All right. The next step here is to take a look and see where, test tube 1 was really this test tube we had right here. So theoretically, you know, this is actually test tube 1. I just didn't put the number on it. So we've got test tube 1 here. We're going to take a look and see if what we can see in it. There's essentially no glowing there. Okay. Test tube 2, which is after the binding buffer was put in there. There does not appear to be any glowing. Let's see test tube 3. Test tube 3, I don't know if you could tell, there is a green glow when you look at it from the side. I don't know if you guys can see that in the camera. So there's a small amount of green glow in there that we've actually separated. And it's hard, I don't know the best background for you to see that in, but definitely a green glow when you look at it. Now, a couple things I need to make sure you understand. Our purpose, very simple, was to get the PGlow protein out of this. We want to harvest the PGlow. So we try to get the PGlow out of the bacteria, and to do that we use this column chromatography to separate it out based on ionic exchange. And then at the very end, when we washed it, we washed all of the solid trapped targeted proteins, and they should wash into this last tube. This is the tube that is a glowing tube, and that tube would be the end result of why we tried to do this, okay? The big question here is, and this is what you gotta, when you're talking to your students about this kind of stuff, all this equipment comes with it. When we give you the kit for this, we supply the syringe with the test tubes, with the single hole stoppers that fit into this exactly. Kind of a key deal, because this way you can actually do this whole lab in 2 days, okay? You can set it up one day, let it incubate, and the next day, even with a freeze thaw, you should be able to get an extraction. Now, how to get good results to where your kids can see really good glowing tubes, if they inoculate these plates when they do the PGlow lab and they separate it out to where you're just getting the glowing bacteria and not any of the white bacteria that's not glowing, that makes it a lot easier to just separate the PGlow from the other bacteria that's in there. So that would be one step that might help. But the purpose is very simple. Can we take a transformed bacterial gene product, and can we separate the product? If this product is for other, this PGlow is just a product that we're seeing to glow. If that product is something that's beneficial to man, this is how we would harvest it through column chromatography. And it's just another biotech tool that we can use to learn a lot about biology and agricultural science. Thank you.